Bismillahirrahmanirrahim. The lecture title is DNA and RNA, Part 1, Nucleotide. Nucleotides is the monomer unit of DNA and RNA. DNA consists of monomer unit called nucleotide. Nucleotides very important unit not only in DNA and RNA, but it is work as coenzyme like ATP, GTP, UDP in the metabolism process. Also, we have nucleotide called cyclic AMP, work as second messenger, and cyclic GMP. It's controlled by ADP in oxidative phosphorylation process, and it has allosteric enzyme regulation activity. Synthetic nucleotide like synthetic purine or pyrimidine analog contain halogen, thiol, additional nitrogen, may use as a drug in chemotherapy of cancer and AIDS, and some immune response during organ transplantation. The next slide, slide number 3, represents the structure of basic nitrogen base purine and pyrimidine. The small name purine for the large structure purine, and the small name the large name for the small structure. We have purine, small name for the large structure, and large name pyrimidine for the small structure. Slide number 4 represents the nucleotide present in DNA and RNA. Nitrogen base, we have 4 nitrogen base conjugate with sugar and conjugate with phosphate group. If the base conjugate with only sugar, we call nucleoside. And when the nucleoside conjugate with phosphate group, will give us nucleotide. Slide number 5 represents the structure of ATP. We have adenine, ribose sugar, and 3 phosphate group. If we have 1 phosphate group, the compound called adenosine monophosphate, AMP. If we have 2 phosphate group, adenosine diphosphate, ADP. And if we have 3 phosphate group, the compound called adenosine triphosphate, ATP. Slide number 5 represents the chemical structure of nitrogen base. We have nitrogen base, adenine, guanine, cytosine, uracil, thymine. If the nitrogen base conjugate with sugar, we have nucleoside. Nucleoside called for adenine, adenosine, for guanine, guanosine, for cytosine, cytidine for uridine, uracil, uridine for the thymine, thymidine. So the nucleotide of adenine called adenosine monophosphate. Nucleotide for guanine called guanosine monophosphate. Nucleotide for cytosine, cytidine monophosphate. Nucleotide for uracil, uridine monophosphate. For thymine, thymidine monophosphate. Okay, this is the structure of nucleoside. If we have nucleotide in DNA, the nitrogen base will conjugate with deoxyribose sugar. So we have adenine and deoxyribose sugar and phosphate group. We have some drug. Can we synthesize from purine and pyrimidine base analog like 5-fluorouracil, 5-iodo, 2-deoxyuridine, thioguanine. This is example of some nitrogen base analog. Okay, in the next slide we have nucleotide used as coenzyme, as supplement, like adenine conjugate with ribose with phosphate. This compound can be synthesized as coenzyme like NAD and NADP and FAD. Very important in the work of the enzyme at metabolism. Okay. So we have nucleotide conjugate with together to form DNA or RNA. So if we have nucleotide like guanine, adenine, thymine, cytosine, adenine, conjugate with, can be drawn as a line represent the sugar. Line, adenine, line represent the sugar, and phosphate group, then to the next nitrogen base, then sugar, phosphate group. They'll conjugate between nucleotide and other with phosphate group. We can draw the DNA strain as phosphate guanine, phosphate adenine, phosphate thymine as example, or we can delete phosphate group, use only the nitrogen base because it's important in the determination of genetic code. The ribose or deoxyribose is fixed, standard. Phosphate group also don't change. The only DNA or RNA, the change part is nitrogen base. So we can write the only nitrogen base represent the genetic code like guanine, adenine, thymine, cytosine, adenine. This is the small example on the DNA strain. The genetic code will convert to the protein, and this protein maybe work as enzyme in the metabolism process. So the strain will call nucleic acid, DNA and RNA, nucleic acid in the nucleus, with RNA in the cytoplasm. Thank you very much for your listening.